I'd like to just get us started really quickly with our very first poll question. So what is your main concern regarding viral vector manufacturing? You've got 4 choices. You've got quality, regulatory, yield, or other. And actually, I would challenge everybody in the audience today for other. If you're going to answer other, please feel free to let us know if, for example, you may have unicorns running through your facility. We'd love to hear about that. What are you challenged by in particular if it's other? I think we have the answers to the poll questions, Miles, if you want to share those and see what folks are struggling with. We did have someone respond to D, other, and the challenge is balancing quality and yield, which I would argue is probably accurate for a lot of folks today. It looks like the main concern regarding viral vector manufacturing is, as we're here to discuss today, quality, with 45.1% of the vote. We've also got regulatory coming in, it looks like in 3rd place with 15%, yield 35%, and other, we've got 3.9, which, as I said, is the challenge of balancing quality and yield to one guest here today. So how do you guys feel about those responses? Would you agree, Andrew and Justin? Do you know, are there areas that you're more concerned about, especially as it relates to, for example, our highest response, quality or regulatory?
Well, if I had to rank these 4 things in percentiles, what I was most concerned with, I think the intelligence of the group is pretty much what I would have said. I would have quality around 50%, and then yield, and then regulatory concerns third. Yield. I mean, I did some calculations on yield as like preparation for this. So, and you could, the interesting thing about AAV is that, well, bioprocess productivity kind of varies on a linear scale, dose and process productivity can vary on a log scale. So at the high end, you can be dosing, say, 1E14 capsids per kilogram, so you could times that by another 7,500 in an adult. And then if you do a very small indication and say the I, you could be dosing as low as 1E11 VG total. So you can have like really log scales in demands. And if you do have a dose that needs, say, 1E13 VG per kg, say a reasonable systematic dose, if you have kind of a mediumly productive process, say at the higher end, 5E11 titer with a 30% yield, out of a 500 liter bioreactor, you're really getting in the range of 30 to 100, maybe 1,000 doses at 1E12 VG per kg, but you're really talking about kind of double to triple digits of doses at the higher end rather than more. So I think yield really is a big issue for the high dose systematic indications, much more so than many other bioprocesses.
Justin, would you agree with that assessment? Is, you know, in terms of the poll and in terms of the math here that Andrew just did for us?
I mean, I think Andrew brings up a good point here. I think it somewhat depends on the program and the indication you're going after, where your biggest concerns are. If you're after a neuromuscular condition, and the doses are very high, you probably have a lot more concern around the yield and potentially around the quality piece of that, because you're going to a very high dose. If you're going for an ocular indication, there may be less concern around the yield. And some of this depends on if you're going to a rare disease population versus a larger disease population where your concerns lie. And so I think each program, as I've been here, Regenx has several programs that have gone through development. We've probably had different concerns on each program as we've gone through there that would get an increased attention. I would note one on here, maybe it's probably on the lower end on the regulatory, but I think it's one that's kept me going as and thinking all the time as we go through these. I think one thing that's unique in the cell and gene therapy field is a lot of, when you're going to the rare disease space, I think the populations can still be quite heterogeneous. You're going after, you're learning science as you go through this, and you're reacting to data directly. So you end up with fairly small data sets, and as far as CMC standpoint goes, and you can also have programs that can go through very quick accelerations whenever you demonstrate compelling data. So It does, I put a little bit of stress on the regulatory pathway as you go through this, because of how do you navigate from something that you're striving to bring a treatment forward to patients, and then you're showing some early success, and then how do you work to accelerate that? How do you do that in how do you bring that value to quickly to a small indication in the space? So that's one that I reflect on. That's one that we've gone through. And also, how do you respond? Oftentimes you may be dose escalating. The dose is uncertain. How do you respond from a CMC standpoint quickly to data in the clinic?
That's been a big challenge, I think, in general. Andrew, did you want to add anything to that? I know, in terms of everybody talking about the regulatory paradigm and how quickly we have to move, that timeline keeps getting shorter and shorter, right? Between your preclinical development and clinic even, and commercialization.
I was going to say, I completely agree with that. And I would be specific in saying that what keeps me up at night with regulatory is comparability. So I know certain processes that have been approved to switch from transient transfection to SF9, which to me is a massive change. And then if you have a program that dose escalates higher than you expected, you need higher yields, you have to change the process. That's the thing that I really worry about in regulatory. And even simple things like, have I stashed all the samples I need in the freezer from these early batches, or can I change the method and now I can't directly compare them? Like these are the kind of things, especially when material can be tight, especially with some of the earlier processes.
I want to stress this is a rapidly innovating field. So oftentimes companies are going through, innovation on their side, and you may be forced with an opportunity for compelling change that can help from a process standpoint, bring, robustness to your process, or scale to your process. And how do you navigate that through when you're rapidly innovating and a rapidly advancing program? And comparability is at the core of that. How do you manage the comparability through that so you can bring that innovation forward?
Absolutely. And that's why we now have regulatory guidance, right? Draft guidance on it. We have conferences specific to comparability. I think there should be bumper stickers, #GotRetains, right? I think that's another, I feel like we need that in this space. So if anybody's watching and has the power to make that happen, please make that happen.
That may not help your yield. You may be more concerned about yield if you're trying to hold a lot of retains too. So that's always
there's that too.
Debate as you go through.
Yeah.